coming off of cells that make them move around. Bye, guys. They are all throughout the cell, again, as I said, to preserve the shape. Also to change the shape, kind of like with the muscles. They contract in some spaces and expand in others. And there's also microfilaments which facilitate transport of stuff inside the cell. So I'm going to go through these one by one. And one thing, because they are so important, they are inside every single cell ever. It's kind of a fundamental necessity, kind of like having DNA. If you didn't have DNA, you'd just be a floating package of proteins. The smallest are actin filaments. Actin you need to know. Or microfilaments. These shrink, they're basically like a line. They shrink at one end and expand at the other. And that's how you have, like, they act kind of like cables that will pull or relax, similar to how muscles work on a larger scale. And this is how it will change the overall shape of the cell to go from, say, a sphere to more of a wobbly shape, kind of like an amoeba, or contract like in a muscle. They can align together, so all of the shrinky ends are at one side and all of the expandy ends are on the other side. And those are polar or non-polar, respectively. So similarly to how polar molecules have all pluses at one end and minus at the other, a polar microfilament has all of the shrinky bits at one end and, unshrinky bits at the other. Next is intermediate filaments. So these tend not to do a whole lot. They're just there to hold the shape. They are kind of like non-pulling cables that just keep the tension of the cell strong enough not to snap. And they will basically hold the shape. Next are microtubules. So these are the rigid support beams of the cell. If the first two are like cables that have tension, these resist pushing forces. They're hollow. They're usually about, I think I miswrote that. But basically they're hollow cylinders. There you go. Yeah, so they're hollow cylinders, and they're actually bigger than some other parts of the cell. And so things will travel inside and on the outside of them. So when you have vesicles moving throughout the cell, most of the time they're not just floating, they're traveling along this with a bit of actin helping them move. Remember, actin will shrink and expand, and that helps it crawl or walk across these microtubules. So they're kind of like the highway within a cell. What? I have a water bottle back there. Can you grab it? I'm almost done. After this, I go on to Nathan. And these, if you remember, are also used by the centrioles and centrosomes during mitosis and meiosis. They grab the chromosomes, pull them, snap them in half. If it's not there, it's fine. Don't worry about it. So again, they facilitate intracellular transport. They hold the shape from pushing forces that would try to contract them. They're used in cell division. They're also used to create the cell wall in plants. Not just the structure, but physically building it. Cilia and flagella use these as kind of like the overall rigid structure inside that gets moved. And they're hollow cylinders. At this point, I'm going to turn it over to Nathan.